Ladies and gentlemen, I would like to welcome you to a discussion with this year's recipient of the Otto Warburg Medal, Professor Maya Schuldiner, molecular biologist at the Department of Molecular Genetics at the Weizmann Institute of Science in Rehovot, Israel, who's receiving this year's award in recognition of her pioneering work on cellular transport mechanisms of proteins and the role of contact sites between cell organelles. Congratulations to this award, Maya. I also welcome Professor Nikolaus Pfanner, medical doctor and molecular biologist at the Institute for Biochemistry and Molecular Biology at the University of Freiburg. He is the winner of the 2015 Warburg Medal for his groundbreaking work in the field of mitochondria. First of all, thank you to both of you agreeing to take part in this interview, which has become a tradition as the prelude of the Otto Warburg Medal award ceremony. My name is Werner Müller-Esterl. I'm a physician and biochemist and a long-standing member of the GBM. Please allow me to say a few words about the award itself. The Otto Warburg Medal has been presented since 1963 in honor of the German Nobel Prize winner of 1931, Otto Warburg. Many prominent figures have received this award, including 10 other Nobel Prize winners, such as Günter Blobel, Randy Schekman, and Emmanuel Charpentier. It is awarded to outstanding researchers and for their work in biochemistry and molecular biology, and it comes with a prize of 25,000 euros. For the past 14 years, Elsevier, a scientific publisher, has sponsored this prize, which is awarded by the Society for Biochemistry and Molecular Biology, and its members also make the nominations. Now let's turn to the winner of the 2026 prize. Maya, congratulations once again for this prestigious award. Could you tell us what significance the Otto Warburg Medal has for you and your team?
Thank you. Yes, first of all, it was a real excitement and huge honor to, when I was notified of the award, I was not expecting it in any way. And I felt that it really gave a huge boost to my sense of the science that we're doing and how much it is appreciated. And so it was a very special moment for me. I would say that on a global level, at a time when fundamental science is often being slowly chewed away from the fabric of the universities, to me, this is extremely important that we continue to value and promote people that work on fundamental scientific problems. And that for me was a huge part of how important this is. It also showcases to our students that there is a value and that it is appreciated to continue and do basic cell biological research, which is important because there's so much still to discover. On a personal level, it's been a trying time to be a scientist in Israel. We're being isolated from a lot of the academic milieus around the world. And so it warms my heart to have this friendship with Germany and the GBM. And it means a lot for us in Israel, for all of Israeli scientists, that we have this long-lasting friendship and support from you.
Thank you. Klaus, you have received many important awards, including the Leibniz Prize and the Walter Neupert Prize. What appeal did the Otto Warburg Medal have for you, and did it have any tangible impact on your scientific work?
Yeah, first of all, congratulations to Maya for receiving the Otto Warburg Medal for her outstanding work. For me, the Otto Warburg Medal was a great honor, not only for myself, but for my entire team, who worked on mitochondrial biogenesis with great enthusiasm. Seeing the list of eminent scientists who received the Otto Warburg Medal, I'm deeply grateful to the GBM and BBA, to my scientific mentors, Walter Neupert and Jim Rossman, to my team, and to my family for supporting me. The Otto Warburg Medal has been very special to me since I received my very first training in biochemistry as a medical student in Munich by Theodor Bücher, who was a student of Otto Warburg. And Walter Neupert, my scientific supervisor and mentor, was a student of Theodor Bücher and thus a scientific grandson of Otto Warburg. Receiving this medal meant for me to reflect about my scientific research, about the findings we have made, and more broadly, about the development of the field. The Otto Warburg Medal was a trigger for opening the mind and for looking forward to future developments of research on cell organelles.
Thank you. Mother, your fundamental research into intercellular and interorganelle contact sites was a key factor in your being nominated for the Warburg Medal. Could you briefly explain the functions of these transient interorganelle connections?
Absolutely. I think they're one of the wonders of cell biology, because if we think of the cell as a small molecular machine, for many decades, it was thought that each part of this machine acts in isolation. But as we know, the best machines are those where all of the parts have integrated functions, and the functions all work together to create one holistic output. And this is exactly what contact sites allow the cell to do. They take each organelle with its unique function, stress responses, and sensing mechanisms, and allow them to come together physically, create physical borders where they can transmit small molecules, information, and physically associate for aspects such as inheritance or quality control. And this basically allows an integration of the entire cellular system and provides the complexity of information inside the cell to be carried out in the best possible way.
Klaus, textbooks typically describe mitochondria as the powerhouses of cells. However, their range of functions is much broader and includes acting as signal transmitters. Could you tell us about the mitochondrial output systems that regulate the activity of other cellular organelles?
Yeah, it's really exciting to see the development of mitochondrial research. I have been lucky to see this development for now more than 40 years, and the number of groups working on mitochondria is growing continuously. In the beginning, the main focus was on the powerhouses, but then numerous lines of research opened a broad spectrum of mitochondrial functions, including metabolism, protein biogenesis, membrane dynamics, signaling, apoptosis, and quality control. And there are many output systems in ways how mitochondria influence other cellular compartments. First, the ratio of ATP to ADP in the cytosol and other cell organelles strongly depends on the activity of mitochondria and will, of course, influence a large number of processes in the cell. Second, redox signaling. Mitochondria generate ROS, the reactive oxygen species, that can be released and function as signal molecules to influence autophagy, inflammation, stress response pathways, and gene expression. Third, the mitochondrial protein import system can function as sensor for mitochondrial fitness. Accumulation of precursor proteins in the cytosol can trigger stress responses. There are sophisticated systems where proteins are partially imported into mitochondria, proteolytically processed, and then released. Under stress conditions, the proteins or their fragments can induce signaling pathways to the nucleus and influence protein synthesis and quality control. Fourth, proteins such as cytochrome C can be released from the intermembrane space into the cytosol and function as trigger in apoptosis. And fifth, you see the list is quite long. And fifth, mitochondria release a factor that is required for the biosynthesis of iron-sulfur clusters in the cytosol. Roland Lill and colleagues showed that this is a reason why mitochondria are essential for cell viability. And there are many more ways how mitochondria communicate with the rest of the cell. I will just name a few. Metabolites for mitochondria can function as signals and influence epigenetic enzymes. Mitochondria can uptake and release calcium and are thus involved in calcium signaling. Mitochondria can release vesicles with different contents. And mitochondria can release DNA and RNA into the cytosol under different stress conditions, then can activate immune signaling pathways. So to sum up, there's a growing number of mechanisms and pathways how mitochondria are integrated into cellular signaling.
Okay, thank you. As previously mentioned, contact points are transient in nature. Which proteins enable the corresponding tethering, and how are they regulated in terms of strength and duration of the interaction?
That's a great question. So, as I said, contact sites are like borders, but in a way they're more like zippers. So if you think about it, although the membranes never actually fuse, this is part of the definition of a contact site, the tethering molecules, the tethers, the proteins that act as tethers, actually intercalate, physically bind each other and allow the physical association between the two membranes. And this can be regulated on a variety of different levels. Of course, the levels of the different proteins that are produced can create more or less tethering. The proteins themselves can be post-translationally regulated. For example, binding can be dissociated by a phosphorylation site that will now not enable a binding interface. And there are also beautiful examples where the interaction is not between a protein and a protein, but a protein and a lipid, or a protein and a molecule on the surface of the other organelle. And this can be regulated by turnover of the lipid or alterations of the head group by various mechanisms. And so depending on these three levels of regulation, we see contact sites that are extremely long-lived. Actually, there are some contact sites that seem to stay forever once they are formed, and ones that are phenomenally transient to the level of even seconds, that can associate and dissociate on demand, depending on the flux of lipids, for example, that mediate them. And so I think the beauty of this is that we have, on the one side, a system that can be stable and define the cellular localization of organelles, or the three-dimensional organization in the cell, as well as their inheritance and their co-movement, so they can be as strong as to enable movement of entire organelles together. And on the other hand, can be kind of rapid kiss and run interfaces where transfer of molecules is created on demand.
Thank you. Following on from the previous question to you, what is the role of extracellular mitochondrial DNA and extracellular mitochondria at the systemic level?
Yes, the detection of extracellular mitochondrial DNA in even entire mitochondria is a rapidly developing area with numerous interesting observations, but also many open questions. Mitochondrial DNA of fragments can be found outside of cells, naked, or in vesicles, or in intact mitochondria. The release can be passive during cell damage or cell death, or active via extracellular vesicles. Many systemic functions are discussed for the role of extracellular mitochondria and DNA, from stress signals and metabolic messengers to transfer between cells and coordination of immune activation, inflammation, and tissue repair, but they may also contribute to chronic inflammation and disease. The area is exciting, but needs mechanistic and molecular understanding in order to properly assess the various observations. I expect that we will read a lot about the impact and the underlying mechanisms in the coming years.
Okay. Another of your favorite topics is the intercellular targeting of newly synthesized proteins. Is the endoplasmic reticulum surface retrieval pathway that you presented so well for the yeast, is that also present in eukaryotic cells?
So can I do a politician's move and take your question as an opportunity to also insert something that I was hoping to say, which is that actually it is this process of protein targeting and translocation that led me, that caused me to be the scientist that I am today. And this is actually thanks to my hero here, Klaus, whose talk I heard. So it's very exciting for me to be able to have this interview together with you, because when I was a PhD student, I was actually working on early human development using human embryonic stem cells. And I heard Klaus give a talk. And this was the first time that I heard about three concepts that really altered my perception of biology. The first is the elegant use of yeast biology, yeast genetics, yeast biochemistry to solve fundamental questions in cell biology, as Klaus does. The second was the idea that you can take a list of proteins that have no characterized functions and use clever experimentation to figure out what they do. And the third, and of course, as Klaus has done over the years and just has slowly figured out many of the proteins that allow translocation into mitochondria. And the third was the mind-blowing idea that the organelles are populated by proteins that have to properly target and translocate into them from the cytosol where they're synthesized, and that this allows not only specialized functions of each organelle, but also regulation at the level of the composition of each of these compartments. And this is actually what led me to look for my postdoctoral studies in a very different field, functional genomics of proteins in yeast, focusing on targeting and translocation. So this is an opportunity to say thank you to you, Klaus, for this inspiration. And I've never looked back. I love what I'm doing. And I love the surprises that one encounters along the way in this process of doing science. And the ER surface retrieval pathway that was discovered in collaboration with the Professor Hannes Hermann is one of those surprises. Who would have thought that when you want to send proteins to one organelle, you would have to route them through another? And this was a wonderful collaboration and an exciting discovery. Is it conserved in mammalian cells? We are still trying to figure out.
Thank you for this detour. Plus, you have studied the structure of the mitochondrial transport and import machinery in great detail, often a very cumbersome undertaking because we are dealing with integral membrane proteins. I would be interested to know whether AlphaFold, the smart method to predict 3D structures of proteins, has already helped to advance your research.
Yeah, the studies on protein transport into mitochondria started with one linear pathway, and Maya, of course, summarized very nicely many points of the developments on this mitochondrial research. Now we have at least 5 different import pathways and a network of machineries. We and all the groups working on mitochondrial protein import want to understand the molecular mechanisms, structure, and function of the transport machineries. AlphaFold is indeed very helpful for our research. We use it quite frequently. Of course, you should have a structure of the transport machinery at high resolution, typically via cryo-electron microscopy. But then for generating mutants, AlphaFold is helpful to plan the appropriate mutants and to screen for potential alterations. AlphaFold helps us a lot to plan experiments on structure function studies. But even when you have a new protein without known structure, AlphaFold is helpful with predictions of the likely structure and thus can guide the next experiments.
Thank you. Maya, contact points contribute to the metabolic regulation. For example, they influence the cellular steroid biosynthesis. Could you please explain what these regulator phenomena entail and which important metabolic pathways they affect?
So I think what we're starting to realize is that, in fact, there isn't a process that this does not impact. Because if we once thought that signals and metabolites diffuse through the cytosol randomly, an enormous body of work now suggests that, in fact, it's very unlikely that metabolites just travel randomly through the cytosol, both because this is not an efficient way to move things around the cell and also because of the timescales that it would take. And in fact, a much more efficient way of moving metabolites around the cell is through dedicated areas of transfer between organelles. And a lot of the metabolic pathways require transition between membranes of different organelles or lumens of different organelles that contribute to various parts of the metabolic pathway. And there are an increasing number of examples where it is these contact sites that allow the rapid transfer. The most beautiful, I think, the first and still most beautiful example was discovered by Jan Vance already in the 80s. And this was the phospholipid biosynthesis pathway that shows the transfer of lipid precursors made in the ER to mitochondria for alterations of the head group, and then transfer back to the ER for continued biosynthesis. And indeed, she could show that this requires these contact sites because the ability to rapidly move molecules back and forth in a regulated manner enables these biosynthetic routes to occur at the rate in which the cell can utilize them. And so this is also true for cofactors. For example, during sterile biosynthesis, as you said, the heme cofactor is an absolute essential requirement, and that is, of course, produced inside the mitochondrial matrix. And so the place where you would want to have the handoff or the transfer of the cofactor that is now required in high amounts when you upregulate this biosynthesis is at the contact sites. And now every single contact site that is being studied more in depth, if it's between the peroxisome and the mitochondria, the endoplasmic reticulum and the mitochondria, the plasma membrane and the lysosome, we're seeing these collaborations between organelles, and these enable the rapid formation of metabolites at a rate that can sustain life.
Fine. Mitochondria undergo constant fusion and fission processes and dynamically interact with organelles such as ER, as we heard, peroxisomes, lipid droplets, and so on, which brings us again close to Maya's work. Is this crosstalk essential for mitochondrial function?
The answer is yes. The fusion and fission dynamics in interorganelle contacts are tightly integrated systems that control mitochondrial metabolism, quality control, and signaling. So here are some examples. Fusion of mitochondria mixes mitochondrial content and can buffer deficiency by complementation. Fission of mitochondria can segregate damaged mitochondria and is required for mitophagy and the distribution of mitochondria during cell division. Contact sites between ER and mitochondria are central regulatory hubs, as Maya explained beautifully. They are required for mitochondrial fission. The ER can wrap around mitochondria and mark the division sites. For lipid transfer, mitochondria receive the majority of their phospholipids from the ER, and also for calcium homeostasis. Mitochondria peroxisome interactions promote metabolic cooperation on redox metabolites and on the beta oxidation of fatty acids. And mitochondria and peroxisomes share components of their fission machineries, such as FIS1. Mitochondrial lipid droplet contacts help in energy flexibility, including fatty acid transfer and metabolic switching. When the crosstalk of mitochondria with other cell organelles is disrupted, mitochondrial ATP synthesis can be decreased, the production of ROS increased, mitophagy can be impaired, and the cells have less metabolic flexibility.
Thank you. Maya, not unexpectedly, contact sites can play important genetic roles. For example, in congenital generalized lipodystrophy type 2, via the tether protein cypin, or in immunological responses via calcium signals at the ER plasma membrane contact sites. Are there already approaches to intervene therapeutically at this point?
So first, I would like to say that for me, one of the most beautiful things about fundamental cell biology is that as a field starts to expand, so 20 years ago, I think there were maybe 10 people all around the world studying contact sites. And these were considered a very, I would say, rare or unique phenomena. And as the understanding that this is actually a fundamental concept in cell biology started to expand, and more molecules started to be discovered, mediating the contact sites as tethers, regulating the contact sites, transferring molecules and signals at the contact site. This created the basic and fundamental knowledge required to now understand what the role in development, physiology, and disease is. So only once we had the molecular players could we start to understand whether those players are important for more complex processes. And so now we're witnessing a golden age where all of these initial discoveries of the important molecules that guide the formation and regulation of contact sites are starting to be associated with the physiology of more, not only of cells, but of more complex organisms. And with this increase in the discovery of how these molecules play a role in development and disease, we're starting to understand that, in fact, many diseases are impacted by contact sites, not only diseases that are specific to contact sites, but also that contact sites contribute to the progression of many of the diseases that are, we see them as common and multifactorial, such as cancer and neurodegeneration. So of course we should be able, and we want to be able to modulate their amount, their abundance, their strength, and their activity. And so there are huge efforts around the world. I was actually part of a consortium that looked into it, that are looking for drugs that can modulate the activity of the various tethering molecules and regulators. And I think the next decade or so, we will really see a huge increase in the discovery of such molecules. And as we understand more and more about the molecular machinery, we will be able to guide these types of searches and experiments even better, to reach, in the end, tools that will allow us to really tweak specific contact sites under specific physiological conditions. And this is up and coming.
Thank you. Over the past decade, numerous mitochondrial diseases have been described, such as optic atrophy or Charcot-Marie-Tooth neuropathy. Are there promising developments for the treatment of such mitochondrial diseases at this time?
There's a growing number of human diseases linked to mitochondria. PubMed lists more than 6,000 publications on mitochondrial disease just in the year 2025. So the current guess is that more than 500 genes coding for mitochondrial proteins are linked to human diseases. And a large number of basic and clinical research groups are working in order to understand the mechanisms and pathogenesis of mitochondrial diseases and to improve the therapies. Since there are many different mitochondrial diseases, there's not one common cure. At present, treatment is often supportive and symptom-focused, involving multiple clinical disciplines from neurology to cardiology, endocrinology, and metabolic support. In a few cases, there are specific disease-targeted therapies, such as supplementation of coenzyme Q10 in patients with primary COX-10 deficiency. But also here, the effect can be limited, and a careful monitoring and further research will be needed. The emerging possibilities for therapies include dietary intervention, small molecules enhancing mitochondrial biogenesis, stem cell transplantation, gene therapy, and RNA-based treatments. Basic research and clinical trials are ongoing, but there are still many critical challenges on the way for obtaining targeted therapies for mitochondrial diseases. So there is hope, but it will take time and a lot of future research.
Sure. To conclude the research section, Maya, could you tell us briefly where you see the focus of future research work and what breakthroughs do you expect for your field in the coming decade?
So I think the field of contact sites has really now evolved to the stage that you alluded to very correctly, the stage where we have a very good understanding of the fundamental principles guiding contact site formation, and we now want to understand how they impact development, physiology, and disease, as well as tailored drugs for them. So I think that you were kind of right on in the assessment of where the field is at. Similarly, the field of targeting and translocation, we have probably not finished, but I think in the last couple of decades, we've mapped the complexity of routes that are important to send proteins to the right destination. And so now the implications of this in more translational research are also up and coming. I do want to say that in the field of functional genomics, which is kind of my core field in the discovery of protein functions, one would think that we would be further along. But despite the fact that we have already sequenced genomes of eukaryotes for nearly 30 years now. Next year it will be, actually this year it's 30 years exactly to the sequencing of the first eukaryotic genome of the yeast. We still do not know what about a quarter of its proteins are doing at all. And many of these are conserved all the way to humans. In humans, there's an even greater number of proteins whose function we do not yet understand. And this becomes, of course, even more elaborate as one goes out of the model organisms and into the natural variation. And so if you want to understand life at its most fundamental ability, we need to know what all the proteins in a cell are doing. We need to know what all the proteins in an organism are doing, and we need to understand the repertoire of protein functions that drive life in all kingdoms. And I think this is a huge endeavor for the next not only decade, but decades to come. And it will be this capacity that will guide also our ability to provide truly personalized treatment to patients, because only if we understand what all of their proteins are doing can we really tailor solutions that will enable them to live better and hopefully longer as well.
Fantastic perspectives. Klaus, going back to you, what new insights in mitochondrial research do you expect to emerge over the next decade? Will we soon have reliable tools for genetic manipulation of mitochondrial DNA?
Let's start with basic research. I expect that we will understand the molecular mechanisms, structure, and function of the major mitochondrial machineries and their integration into membrane-spanning networks. We will get a deep understanding of the role of mitochondria in cellular signaling and quality control. We will get insight into the organization and function of mitochondrial networks with other cell organelles, as Maya, of course, explained beautifully, in different tissues, under stress conditions, and in development. System-level research and AI may indeed help to better understand and possibly also to predict mitochondrial network behavior in health and disease. The impact and mechanisms of extracellular mitochondria and DNA will surely be an important task. And as outlined to the previous question, we can expect that targeted therapies may become available for at least some mitochondrial diseases. And the role of mitochondrial decline in aging will become an important research topic. Regarding the mitochondrial genome, it encodes only 1% of the different mitochondrial proteins. but is involved in many mitochondrial diseases. In humans, the mitochondrial genome codes for 13 proteins with important functions in the respiratory chain and ATP synthase. Many attempts have been made to find tools for the specific manipulation of mitochondrial DNA. The efficient delivery of specific oligonucleotides into mitochondria is one of the major hurdles. Very recently, Peter Rehling and his team reported an exciting breakthrough. They established a system to efficiently import morpholino nucleic acid analogs into mitochondria. Antisense morpholinos specifically block translation of their target mRNAs, thus enabling targeted silencing of mitochondrial gene expression at the level of mRNAs. I expect that within the next decade or so, efficient tools for genetic manipulation of the human mitochondrial DNA will also become available.
Yeah, thank you, Maya. And I have a few more questions to you. You have worked at various institutions and that have shaped you and your research. What advice would you give to young people, early career researchers that start out on a scientific career?
I have two pieces of advice. The first is always look for the best mentors. In my experience, it's even more important than the topic that you choose to research. I was phenomenally lucky to have an amazing mentor during my undergraduate and graduate studies, Professor Nissim Benvenisti, that taught me everything that I know about how to do science, how to be passionate about it, and how to mentor the next generation of students. I owe him so very much. Then during my postdoctoral studies, I had the amazing privilege to work with Professor Jonathan Weissman, who taught me how to think big, conceptually outside the box and make breakthroughs in scientific research. And my life wasn't the same after I came out of his lab. And when I joined the Weizmann Institute, the late Professor Benny Shiloh agreed to be my mentor as I was starting up as a principal investigator. And it is thanks to his wisdom and good advice that I managed to create the type of lab that I had hoped for and wanted. So I think that great people along the way are always the driving force that allows us to flourish. And the second piece of advice is also about people, and that's find great collaborators. And so for me, one of the huge joys of doing science is being able to work with people that I love to interact with, that I'm excited about talking science and throwing ideas about with, and many of which have become extremely good friends over the years. And it is a huge privilege to be able to combine these friendships and our passion for science together with shared discoveries and shared scientific knowledge. And so for me, the road is really about the people that walk along it with you. Maybe if I'm allowed to say a third piece of advice, that would be choose the right partner, because I also couldn't have combined my career with my 3 amazing children without having the most wonderful of partners. My husband, Professor Schuldiner, is actually also a scientist here at the Weizmann Institute of Science, and our friendship and shared parenting of our children throughout the years and shared decisions in every step of our career really allowed me to be the type of scientist and mother and person that I am today.
Thank you. The reform of assessment of scientific achievements is currently the subject of heated and controversial debate in science policy. Klaus, what criteria do you use to select your closest colleagues?
For selecting new professors for the faculty in our institute, don't rely too much on metrics, but a personal assessment of the quality and originality of the publications and the impact of their work on a field and its future development are very important. And of course, talking with the candidates personally, seeing their competence, their personality and scientific thinking are crucial points for the selection. And for selecting new co-workers for my own team, I am not doing this alone, but together with my team. Candidates give a presentation and then talk with many lab members individually. Only when the majority of co-workers is excited about the candidate, about the scientific competence, and also about the social skills, then we have hired a candidate.
Along the same lines, Maya, with all your experience in science, what 3 personality traits do you consider essential for success in research today?
Wow. So I would say, of course, in addition to just being phenomenally passionate, which I think is part of what defines us as a scientist, I would say my 3 favorites are high frustration tolerance. I think as you spend more and more years in science, you realize that if you can't deal with the frustrations of always being on the boundary of knowledge and hitting on brick walls the entire, the majority of the time, it's very hard to stay happy and productive in this field. The second I would say is creativity. So we often hear that maybe AI will do everything that we do and even better in the next years. But I think creative scientists will never be replaced, because the capacity to think outside the box and to come up with technologies, methodologies, ideas, and hypotheses that nobody's ever thought of before is something that the human brain will continue to do better than any machine. And the third, and my personal favorite, is being compassionate. I think science is a community endeavor. It is something that we should be doing as an entire humanity to advance our chances of having a sustainable future. And because of that, we need to develop compassion to our students, to raise them to be great scientists themselves, to our colleagues as we judge their work and give them feedback in reviews, in grants, in manuscripts, at conferences, and to ourselves, and to have compassion for the mistakes that we all make. If we don't find this compassion and spread it around, I think we won't be able to create the amazing community that we deserve to have to drive science forward for the benefit of humanity.
To put this question another way, what do you consider to be the most biggest obstacles to developing a productive research personality class?
Okay. So first, I would say don't think too narrow. Be open for new developments and findings. In the laboratory, you often get unexpected results. Some of these results are likely not useful, but some can be really exciting. So having open eyes for unexpected findings is very important in order to broaden your research. In English, you have this wonderful word, serendipity, for the lucky moment when you find something new and exciting. Second, don't spend too much time for planning your scientific career. What is most important is to be excited about science, as Maya also said. You have to love science, and it is fun in trying to solve a problem to get new biological and mechanistic insight. So in my view, being excited about science, creativity, endurance, and communication are the crucial points for making a career in the life sciences. Third, the external influence can impair science. Formalities and the increasing bureaucracy can indeed pose limitations. But when you love your science, it will be possible to overcome those hurdles. For the politicians and the funding agency, this means they should give the scientists more freedom. Trust the scientists. Then you will get the best outcome from the funding money.
Okay, thank you. Maya, many scientists are concerned about the way research funding is developing, not only in the United States, but also in Germany and other countries. Are you worried that anti-research sentiment could gain ground in your country as well?
Absolutely. So I think there are two main worries. The first is that there's less funding being diverted into science because of certain leaderships not believing that science is an important way to drive humanity forward. And the second is that a lot of the funding is being routed more and more for specific outcomes, for finding cures for cancer, for studying specific types of neurodegeneration. And what we've come to learn, I think over the last couple of decades, is that actually some of the most important discoveries come from curiosity-driven science, from science that is done without a prior idea of moving towards the therapy, but out of the understanding of the molecular mechanisms of how fundamental biological processes occur. And so I would love to see a future where not only is funding plentiful, not too much. I think it's good that it's slightly restrictive, that we have to fight for it and be creative to get it, but enough to sustain high levels of science, but also that is provided to support people's curiosity-driven science and not with an end goal in mind. And unfortunately, so I would actually say, fortunately, in Israel, the Israeli Science Foundation supports fantastic fundamental science, and this is a great attribute of Israeli scientific funding. And for this, I'm extremely grateful. But unfortunately, the Israeli government over the last couple of years has been impinging more and more on the academic freedom of academic institutions in general. And this is a very worrying trend that a lot of us in the Israeli academia are actively fighting against, and hoping that we will manage to stop this worrying trend.
Yeah, thank you for your openness. Plus, the integrity of research is a growing concern, as it's increasingly becoming compromised by a flood of fraudulent papers, AI-produced articles, non-peer-reviewed preprints, paper mill products, and predatory journals. Should scholarly publishing submit to external independent oversight to certify the quality of journals and publishers?
You address an important point. It is a growing problem that some strange publishers and journals don't follow the rules of scientific integrity. We indeed have to think about measures to keep the high standards of scientific publishing. An independent system that will certify the quality of journals and publishers looks like a very interesting option in order to help scientists and funding agencies to select the proper and reliable journals.
So thank you very much to both of you. That brings us to the end of our interview. Congratulations again to you, Maya. I'm looking forward to the Mosbach colloquium in March, where the Otto Warburg Medal will be presented to you. So see you then. Bye-bye.
Thank you so much. Goodbye.
Bye-bye.
Thank you very much to
Great
both of
seeing
you.
Klaus.
Thank you. It was real fun.
Bye-bye.
So stay tuned, please.